All right, good afternoon. You just watched part one that was actually recorded in January of this year, so I'm probably wearing a different shirt. But it's the same background as what we're doing because we're doing this again, but we are investigating new proteins this time. And so now I'm going to tell you what we found in 2024 led us to what I'm doing, what we're doing, and what I set up for you to do. We have a colorful mystery that has only deepened the more we've looked at it because we keep getting interesting colors and don't know for sure which color's which. Let me quickly check to make sure I'm recording right, and we are good. Okay. So just to recap, we're dealing with siderocalin immunoproteins, and if you look at the charges on the surface, you have this blue charged cup that binds red charged things. One of the red charged things is enterobactin. We have a crystal structure. You've already looked at that in the lab. And so I just want to mention that that's all there. But what we did is, the question that we have is, we have this human siderocalin. There's siderocalins in pretty much every mammalian species and fish species. Where do they go? Where do they do? What do they bind? Is our biggest question. Do these have the same shaped cups or different shaped cups? And so what I did is I did a big analysis of 5,000 sequences that were all in the genomic databanks of things that have been sequenced. And I ran that, and you see I got these different groups. These are all lipocalins, not necessarily siderocalins. That means that they bind things that have something like a lipid on them somewhere. Basically, we don't really know exactly what they bind. And some of them bind things like retinoic acid, some of them bind things like enterobactin. And so when you take all of the related sequences, you get these groups, and one of the interesting things is that big group in the middle, that blue group with has several subparts to it, and that has one DFV in it. So let's take a look. If we zoom in on that blue group, we find that it basically has all these different subgroups that I've sort of taken, and the ones that we have information for from the protein data bank I have made big. We have structures for the big things, but you see we don't really have representatives. I mean, this is about maybe 500 different genes, and we have maybe a couple dozen structures, maybe a dozen structures actually. Not nearly enough coverage to really tell what's going on. That's why it's nice to have homology modeling, so we can take a gene for which you don't know the structure, find a structure that's similar, swap in the amino acids from our gene of interest into the structure, and then sort of shake it around a little bit to let it find the lowest energy configuration so it's not like trapped in a weird twist or something like that. The other interesting thing is, as I mentioned, there's enzymes in this group right in the middle, and the ones that are yellow have an EC number, which means that you have evidence that they actually act as an enzyme and are not just a binding protein. So we number these, and human siderocalin is way over there. The enzymes are in that group in the middle. I labeled that group 2. And then you have the other thing I talked about was the chicken XFABP, fatty acid binding protein, and fatty acids are a lipid, so that name actually kind of makes sense. But the chicken structure that we have is up there in the corner in group 5. We also have group 1, 4, 6, and 7 down at the bottom. But mostly we are working this time on groups, we've worked on groups 1, 3, and 5 the most. Mostly groups 1 and 3, actually. But just notice that the enzymes are right in the middle. Maybe there's a way for something to be evolving an enzyme function. And if you look at the different things, we have 3A, which was pink in one set of cells and blue in another set of cells. And then we have 5F, which was blue in one time, but that was a long time ago, and we haven't been able to replicate that. In fact, we've replicated the blue color another way. So if all that is weird, just the most important thing is groups 1 and 3. Group 1 down here is where we have yellow proteins that we got. So we're getting proteins of all these different colors. The human siderocalin is announced in the literature to be orange, right? And so we actually have replicated that nicely by doing exactly what they did, and we have their protein, and it's faintly orange. The thing we didn't expect to get yellow, blue, or pink proteins. So when you do a multiple sequence alignment, and actually this is an easy program to use called CView. We haven't used it this year, but it's very easy to pull up and use if you ever want to do a sequence analysis or alignment. And similar types of amino acids are colored similar colors. And if you can look at this, you see how you have group 3 proteins that are on the left you see a 3, and then on the left you see a 2 down below. And this just shows you, I just took the group 3 proteins with a couple of sequences from structures, and then I took a couple of group 2 protein sequences, and we just threw them into the sequence alignment and said align these and see how they group together. And it still grouped together. The group 3s were all together. The group 2s are all together. They have similarities in the sequences. And in fact, you can even try to see, you can see the dividing line around the middle where you switch from group 3 to group 2, because all of a sudden you have different colors of sequences at the bottom, and those are all group 2. Different colors of sequences at the top, those are all group 3. This sequence similarity is real. You can confirm it by looking at it. And there's enough interesting differences within the group that you could possibly have different functions or binding within the group. These are about 20 to 30% identical. Remember that from that we can tell that it's got the cup structure, almost certainly, but we're not sure what the inside of the cup is. So in 2018, we went through and we recreated the human sideracalin. And the one thing we did that was different from what the literature said is we actually used both DH5 alpha and BL21 cells to make the protein, and then we mixed them. And so I think that we got a fainter orange color, and I replicated it better last year. But we also got 3 surprising colors. We got yellow, pink, and blue. And we found out that, the 1C, we've been able to replicate group 1 proteins tend to come from fish and tend to be that bright yellow. Group 3 proteins, when we've replicated them, actually turn out to be blue. And I'm beginning to think that the pink color, which we've never been able to replicate, is actually a mixture of BL21 cells and DH5 alpha, which we used that time. And I think they're possibly orange and blue mixing together and making the pink color. It's just a hypothesis until I replicate it. But the main thing is we go with what we can replicate. The other thing that we replicated is that we made the blue color again. We made it with the 3A sequence and DH5 alpha cells alone. And so I decided to try to replicate that again this year with our experiments. So that's why we're doing all DH5 alpha, and you might notice we're doing all group 3. I'm hoping to replicate that blue color. We've done it twice, and I want to see how far we can push it, how many different group 3 sequences made in DH5 alphas we might be able to get this blue color from. So we first started to replicate back in, actually this was reversed years. In 2019, we replicated 2018's yellow protein twice. We made more fish proteins, group 1 proteins, and they were all various colors of yellow. There were some of them that didn't bind anything, but they kept binding stuff. And the thing is, those would work in the BL21 cells, which is cool. So this is a list of all the proteins that we've made and the colors that we have observed. And the main thing I want you to see is look at the bottom where we really replicated things. We got orange human sideracalin in winter 2024 when we made them in the 2-star means DH5 alpha cells. And we made some more yellow fish proteins. Those are the group 1s, and those are 1-star. Those work in BL21 cells. And you see that we replicated those the previous 3 years. We got yellow proteins all involving BL21 cells at one point. But the thing that we did different this year is we made 3A protein with only DH5 alphas. And then we got the bright blue. So that's what we're trying to replicate again. And we also tried to replicate the protein that we got before, which is 5F. We did it both ways. We didn't get color either time. So we can chase that down, but it's better to chase down what you can replicate rather than what you can't replicate. So that's what we're trying to do. We're actually directly replicating the 3A result. The 3A is from Cavia porcellus, which is the guinea pig. And each of these has the label, the 3-letter label, and then it has the species name and as an abbreviation. I just do that to basically make sure the labels are different enough that even if the label's a little smudged, I can still tell which protein that is in that tube. Labels can get smudged. So that's where our 6 proteins are coming from. We have 1 protein we're replicating and 5 proteins that are brand new from different species. So when I take all the proteins that we've done, and I also throw in, I throw in a lot of the proteins from a lot of different groups, and we did groups 1, 5, and 3, which were shown on the previous slide, and also group 2, a few examples of group 2, really one example of group 2, but that's one that we know is an enzyme. And so I threw that in, and once again, the sequence tree that is built from those sequences groups together the group 1 grouped together at the top, the group 5 in the middle, and the group 3 grouped together at the bottom. And just remember, these are the examples of the fish tend to be yellow. I don't know what that is, but it's pretty cool. A lot of things can be yellow. Not a lot of things can be blue. And I'm wondering, could it be the blue form of iron that we talked about? Well, we'll have to do some experiments, and we're doing experiments in other classes to find out. In this class, we're making more proteins to see what we get. So just remember that we have the group 1 is yellow. Group 3, we have the one example that's blue. And group 3 also includes our orange human sideracalin. So we might get orange. I don't know. I don't know what we're going to do. So it's kind of fun to see. The other thing that you'll see on this tree is you'll see some things that have the 4-digit or 4 letters, 4 digits and letters, the PDB ID code. You can see 1 DFV and 3 CMP down at the bottom. You're familiar with those. And I just want to mention the other ones I mentioned. 2 WWP is on the bottom of our group 3. You see it at the very bottom of this. And 3 SAO is actually on the top of group 3. There's a couple other PDB IDs that showed up. I put the sequences in and they showed up here. Those might be interesting to look at. I'm not requiring anything, but I'm just saying there are open structures that we could use a pair of eyes on, and if you see something that you think might explain what these things bind and why, definitely let me know. So there's our enzyme. Okay, so I'm going to zoom in on group 3 because that's all that we're doing this year, and I just want to note the PDB IDs that are similar to this, but really we've got human sideracalin right in the middle of things, right next to the 3A protein that we made the blue last time. So it's kind of cool. Let's see what happens. I don't know if we're going to get blue or we're going to get orange or we're going to get pink as a mixture of the two. I don't know. So let's find out. And just notice that as you're doing your structural investigations, you might come up with one of these PDB IDs on your VAST search. That's one of the steps you're asked to do. Keep your eyes open because that's how you'll see something that you don't expect. I'm going to ask you to do agriscan, which basically takes the protein structure and runs an analysis on it of patches that are on the surface that could aggregate, that are polar, that they won't aggregate, or maybe they're non-polar and they will aggregate. And notice that I did the 1DFV dimer. I'm going to ask, I'm asking you to do your monomer, and so I want you to sort of compare it to this. Notice that the 1DFV dimer has this interesting red part right on the lip. I don't know exactly what that's doing. And I also don't know if you take the monomer, do you get a part that is red right where the dimer is coming together? That's the two questions I have when I look at this. And also I look down in that pocket and I see a pocket that is not exactly polar, but it's not aggregation prone either. It's white when you look down into the pocket. So look at your model and sort of compare it to this. And just remember, when you are orienting things, you're going to put the lysine 125 at the 12 o'clock position. This is a cartoon diagram of 4 sideracalins lined up, and they're all oriented. Once you align them with the program, they're oriented so that this one amino acid overlaps. And if you put that amino acid at the top looking down, we'll all be looking down into the calyx the same way. The other thing I'm asking you to do is to do measurements, and this is what a measurement looks like on my old version of PyMol. The important thing is that you have a line showing the exact 2 atoms that are being measured, and then you have the angstroms reported in angstrom units. So I want you to take pictures like this. This picture doesn't have much context, but it's good for showing the exact relationships between those 3 atoms. You decide what you include and what you don't include, but my main thing is make sure you zoom in. Because a lot of times I can only tell so much. If you just show me a picture of the whole protein, then I'll say that looks like a lipocalin. Great. That doesn't tell me anything I don't already know. You have to zoom in to show me details. So the measurements on the old one are done with a wizard. I don't know if it works on the new ones, but I do know that there's a whole community that measures a lot. There must be tutorials out there, and I will do what I can to change the protocol online. As you go through, we have a link to a Google Sheet. You should have edit ability. You should go and find your column. So scroll over to your column, and just with that column there, go down and fill in the colored boxes. There's each of the boxes is right here, and sometimes I have text that goes over into other boxes, but you know how spreadsheets work. That text is still there even if you have to type in the boxes right next to it. Please stay in your own column and don't go into anybody else's column because we need to each stay in our lane. But once you do that, we'll have a whole data sheet, and you're allowed to look at what other people have done as well to compare your protein to them. At the very end, I'm going to ask you to report your distances in the column in those ways. And at the very bottom, I have long questions that, of course, they spill over their box, but you just type in your own box anyways, and it will all coexist. And so just see that we have four questions to kind of get you ready for the final answer. And then I want you to make a call. Do you think that your protein binds enterobactin? Or does it bind enterobactin, a version of enterobactin that looks like enterobactin in two of the pockets but doesn't look like it in the third pocket? Things like that. I don't know what you're going to find, and so that's why I want you to be doing it. So next, a week from now, or 10 days from when I'm recording this, but probably a week after you're watching this, we're going to be back in lab again. We're going to lyse our cells. We're going to run columns. Then we're going to run SDS page gels the next week. And then week 6 is the week where we really have fun. I'm going to set you loose to experiment on your protein. And finally, week 7 is sort of the cleanup week where we are setting up crystal trays. And then we are putting everything in my hands so I can get it ready for future projects. And of course, you're welcome to continue researching on your protein that you've worked so hard to make. So in week 6, we're going to test the protein. We're going to try different ligands, and we're going to see if it makes it change color. So if we get proteins that aren't colored, it's actually okay, because that means that they're probably empty, and that means that we can try to put color in them. So if it's colored, we're going to try to see what happens if we take the color away. If it's not colored, we're going to try to add color by adding ligands. And you want to be thinking about these different kind of structures. Retinoic acid, iron and catecholate rings, which are the rings in the enterobactin, heme, those are all different shapes. We have lipocalins that bind all of those, and I'm very suspicious. For example, I think retinoic acid is usually yellow in some cases. Maybe that's what we're binding in the yellow fish proteins. Are we binding a version of heme that's like metheme when it comes to the blue? Let's find out. And you can come up with a hypothesis, and maybe we can even dig up a chemical that you can try to see in week 6 whether it binds your protein. So we're going to try some more experiments, especially these are even integrated into the class for the Chem 3410 Survey of Physical Chemistry. You can actually take part in independent research alongside with the students who are taking that class for credit. We can talk about that, but first let's make the proteins and see how they work. I want to reiterate to you how new this all is, but that means it's more important for you to be very precise in how you follow the protocols and how you label things, because we're going to be using these labels, and we want to make sure that the proteins are the proteins we think they are. We have ways to replicate to make sure, but when you try to replicate and you don't, that's where you waste a lot of time in science. So let's try to make sure that you label, make sure you're safe, make sure that you just go a little extra on all this, because it actually really matters. This is not just a regular lab exercise. It's a genuine experiment, and I look forward to finding out what you're going to see. All right, thanks for listening. Work in your computer lab this next week, and then I will see you back in lab the first week in November.